The first line of defense. The first line of defense consists of chemical and physical barriers that prevent pathogens from entering the body. An example is skin, which is a physical barrier, preventing pathogens from entering the body. Stomach acid, a chemical barrier, which dissolves pathogens, rendering them useless. And a microbiological barrier of vomiting, which removes harmful pathogens from the body. These are just some of the many defenses that make up the first line of defense. Once the first line of defense is breached and pathogens enter the body, for example, by damaged or cut skin, it triggers the second line of defense. Damaged or cut skin causes necrosis, which is a form of cell death. The second line of defense, or innate immune response, is a non-specific response, meaning anything that is identified as being non-self is a target for it. The innate immune response is activated by the presence of antigens, for example, pathogens entering through any open wound. Some of the first defensive cells that act on pathogens have breached the first line of defense are phagocytes. These cells engulf and destroy pathogens. Cells that are the first to respond once the first line of defense is compromised include neutrophils, phagocytic cells which engulf bacteria and release enzymes to kill that bacteria. Throughout this process they die. These dead cells build up, causing pus. Macrophages, which are phagocytic cells which can digest a large range of infectious agents. Monocytes, found in the bloodstream, which differentiate into macrophages once they leave circulation. Natural killer cells, which travel through the blood and lymph to detect cancerous cells. When a cell becomes infected, it can stop making MHC1 proteins. The natural killer cell, for example, a cytotoxic T cell, detects this as a defective cell, promoting apoptosis or programmed cell death by attaching to a receptor releasing apoptotic-inducing enzymes. Most cells have MHC1 markers, however certain cells have MHC2 markers, found on dendritic cells, monocellular phagocytes, B cells, and macrophages. These cells are regarded to as antigen-presenting cells. These cells, when engulfing a pathogen, will present the antigens of the pathogen to the immune system, preparing other cells to act upon pathogens presenting these antigens. Other cells include mast cells, which have granules that upon contact with non-self antigens release histamine, thus promoting the inflammatory response, and cytokines, which are signaling molecules released by cells such as interferons, which warn cells of incoming pathogens, and interleukins, which assist to stimulate the inflammatory response. The inflammatory response helps contain pathogens by releasing histamine, which promotes vasodilation. This increases blood flow and capillary permeability, which increases the influx of phagocytic cells that move throughout the bloodstream. This inflammation attracts phagocytes and lymphocytes to the site of infection. Another part of the second line of defense involves the complement system. The complement system involves complement proteins, a group of proteins that work together to attract phagocytes and the destruction of pathogens. The complement system assists in an inflammatory response as opsins stick to the bacteria or pathogen, flagging it to phagocytes, attracting them to engulf the cell. This is known as opsonization. A ring of opsins can create a membrane attack complex, or MHC for short, which puncture a hole in the pathogen, allowing fluid to enter, causing the pathogen to rupture. The third line of defense, or adaptive immunity. The lymphatic system is a network of lymphatic vessels containing lymph which reaches all tissues of the body and lymphoid organs. The lymphatic system collects tissue fluid leaked from the blood vessels and returns it to the heart. Fluid is transported throughout the body through lymph vessels which carry leukocytes, white blood cells that fight infection. The thymus is a primary lymphoid organ, which is the site of T cell maturation. Secondary lymphoid tissue, i.e. the spleen and lymph nodes, are important as they are the site of B and T lymphocyte activation. B and T lymphocytes, as well as phagocytes, are antigen-presenting cells, or APCs, which contain MHC2 markers and are found in large numbers within the lymph nodes.
There are two components in the adaptive immune system, the humoral and adaptive immune response. They work separately and together to prevent disease. The humoral immune response is active against non-self antigens outside host cells, for example, some bacteria, viral particles, and free antigens. When antigen-presenting cells phagocytose a pathogen, they present the foreign antigens on the MHC class II markers and migrate to lymphatic vessels. Here, corresponding naive T helper cells use their receptors to recognize the presented antigens on the antigen-presenting cells. This process of selection is known as clonal selection. Simultaneously, free antigens will bind with the specific naive B cells. These are also known as being selected.
When the selected B and T cell locate each other, the B cell presents its antigens to previously selected T helper cells. The T helper cell then releases cytokines, causing the proliferation and differentiation of T helper cells into active T helper cells, including memory T cells. The B cells produce plasma cells and memory B cells. The plasma cells secrete large amounts of specific antibodies which flood into circulation. These are known as free antibodies. The memory cells retain memory of the antigen and specific antibody. This means the body can respond quickly to another exposure to a specific pathogen presenting those specific antigens. The antibodies produced are comprised of two heavy chains, each attached to a shorter, lighter chain in the shape of a Y. The variable region is at the top end of the light and heavy chain, forming the antigen binding site. These antibodies bind to antigens via opsonization, marking them for phagocytes to engulf. The cell-mediated response is active against non-self antigens located inside cells, for example, viruses and cancer cells. When infected, cells present viral antigens or stress-induced antigens on their MHC1 markers. A cytotoxic T cell will bind to this antigen. Coupled with the help of cytokines produced by T helper cells, they proliferate and differentiate into active cytotoxic T cells. Active cytotoxic T cells then recognize the specific infected/cancerous cells and binds to death receptors, initiating a process of cell death known as apoptosis.